Good afternoon, everyone. I hope you are doing well, and thank you for your patience. So, on behalf of Advent Informatics, I warmly welcome you to today's insightful webinar on a highly relevant and cutting-edge topic, which is the Giga-Scale-Compound Library challenges: how GPU and AI are transforming screening. We are delighted to have you with us today and look forward to a session filled with a valuable insight, real-world applications, and interactive learning. Before we begin today's session, allow me to briefly introduce the organization behind this event, which is Advent Informatics Private Limited, headquartered in Pune. Advent Informatics is a leading distributor of advanced life science software solutions. Our expertise spans across chemoinformatics, molecular modeling, medicinal chemistry, chemical data curation, and biological information management. We serve a wide range of clients, including pharmaceutical and chemical companies, contract research organizations, which is CROs, academic institutions, and government R&D labs across India. Our mission is to bridge the gap between complex scientific challenges and advanced software solutions by enabling researchers and world-class technology for efficient discovery and innovations. We proudly collaborate with globally recognized technologies partners such as Molsoft, Chemexon, Elsevier, Campus, Certara, Transgene IT Solution, and KeyModule. So it's now my pleasure to introduce our speakers for today. Dr. Shamsher Singh Sardar, a senior application scientist at Advent Informatics Private Limited. Dr. Sardar is a seasoned expert in computational aided drug design with over 20 years of experience in pharmaceutical and life science industry. He holds a PhD from Jawaharlal Nehru Technological University, Hyderabad. He has held a key scientific role at leading contract research organizations by extensive to drug discovery research. Currently at Advent Informatics, Dr. Sardar is specialized in applying advanced computational tool and AI-driven methodologies to accelerate screening and drug discovery workflows. With this deep expertise, he is the perfect guide for today's session on the Giga-Scale-Compound library challenges, how GPU and AI are transforming screening. During this session, if you have any questions, please feel free to type them in the chat box. So please join me in welcoming Dr. Shamsher Singh Sardar. Dr. Sardar, the floor is yours.
Thank you, Shailesh, for your nice introduction.
Yes, sir.
So, are you able to see my screen?
Yes, sir.
Thank you. Good afternoon, everyone. So, Molsoft ICM, ultra large library screening tools that are available from ICM Molsoft. So, before I start going directly deep diving about the ultra large screening tools, we would like to understand what exactly Molsoft is and how old is the company and what are all the technologies that are being developed, the complete journey since its inception, right? Molsoft is not a new company. It's a 32-year-old company based at San Diego, California. And, the code was written before the first release of the Autodoc. And the earlier adopters of this technology include BMS and Novartis. Molsoft outperformed various commercial and open source tools regarding to the docking accuracy, pros prediction, RMSD, and binding affinity as well. And and AstraZeneca has published a comparative study where they took a set of 168 crystal structures where they redocked, and this in the extreme right where you can see, right? So they compared with Glide, Flexex, and Gold, and ICM outperformed with regards to the RMSD as here. And with regards to the citations, there are more than 6,500 citations in Google Scholar and 200 plus hits from Nature journals and more than 1,300 plus hits in Science Direct, Springer 650 plus, and more than 300 plus in ACS journals as well. And who are all the users? The international users you can see. And this is a very, very small subset what you are seeing here. Pfizer, Novartis, Lilly, AstraZeneca, New York University, NCI and NIH uses MolSoft. And coming to Indian CROs, Syngene, Aragen, Zydus, Origin, PI, and many prestigious institutes from India like Naipar Mohali, Naipar Guwahati, Geetams, and Kakatiya University are using MolSoft for their drug research. This is with regards to the virtual screening. So by the deep integration of AI and the GPU utilization, MolSoft is steering away the drug discovery completely. So MolSoft has a fastest 3D docking tool available, what we call it as RIDGE, Rapid Docking GPU Engine, which can screen up to 10 million compounds in 24 hours on a single GPU. And the benchmarking has been done using RTX 4090. And regarding more details about these RIDGE, we will be looking in the upcoming slides. Again, this is not a synthon-based, means not a fragment-based docking. Again, this is full 3D brute force docking using RIDGE. Again, RIDGE is a structure-based. Coming to a analog-based or the ligand-based, we have something called RIDE, Rapid Isosphere Discovery Engine, which is a pharmacophore-based screening technology, which can screen up to 1.5 million conformers per second per GPU. So in a minute, it can screen up to a database of 1 billion compounds. And we recently published this particular article, which is where this study was done using RIDGE and RIDE on some very tough targets, which we will be looking in the upcoming slides as well. And we also integrated an artisan AI-based scoring function to the MolSoft, and we have released a white paper on it. And we also have something called GigaScreen, which is a deep learning model where you can use 1 billion compounds in 2 days using a normal RTX 4090 GPU. And this is a CombiRIDGE, which is a fragment-based technology where you can consider around 28 billion compounds that can be docked in less than 5 days. And we have something called ligand aid integrated very recently, ligand aid, which is a de novo generation and an artificial design. So this is an AI driven approach where the drug molecules can be directly modified inside the protein binding site. Let's assume now you start with a docking with a small fragment and an initial output. So the dock fragment is extended by adding different R groups on a particular positions using group gen, which is again a neural network technology of the MolSoft. After each round of extension, the newly generated molecules are docked again and see how well they fit into these. So these designs are refined through multiple evolutionary cycles, and this particular iteration may go up to, depending on the active site, maybe up to 3 to 5 iterations. So what makes this tool very, very powerful is the selection process as well here. So at every single stage after optimizing, after the first generation molecules are generated, the weaker designs are filtered out based on the RTCNN score or the drug-like properties and the Moleskin score, which is a synthetic feasibility score. So only the best compounds move forward during this methodology, ligand aid. And I said group gen, which is again it works on a encoding and a decoding kind of technology where in a pocket it puts a vector on a particular area and identifies the nature of the pocket, and then what are the distances between that, but from that particular point what are the different interactions that can be captured. So this way this group gen works, and then those encoding happens, then the decoding is placing them with the suitable R groups. Again, optimization in the active site takes place. And we have something called GigaScreen, which can do a substructure search of 80 billion enamine space and 140 billion Cambridge freedom databases. Just you can do a substructure search and get the results in just 5 to 10 seconds. And we have a neural network based conformer generator tool called Ginger, which can generate up to 10 million compounds conformations in a single day. And we have also recently integrated and neural network-based PK prediction, AI models for mole screen for ADMET prediction. It has around 2,500 high-quality 2D fingerprint, 3D pharmacophore, neural network regressions, neural network uh, classification models in this, where you can use them for the ADMET as well as target identification, drug repurposing, and many more stuff. And we also integrated AI-based torsion profile prediction in the ICM. So yeah, these are all the highlights of what the MoleSoft has. And again, getting back to the Advent, so a single slide on Advent, what we do. So Advent, we were founded in the year 2016, and we are based at Pune, and we are a prominent distributor of life science software databases, drug discovery software, drug discovery and chemical materials, and other services as well. So what do? We provide the first level of technical support to all the products which we distribute, and we were highly appreciated by our clients with regards to our responsiveness here. And 40% of our staff are the PhD holders, right? Using MoleSoft, you can do all kinds of structure-based and ligand-based studies. Coming to Elsevier, we have recently integrated with the Elsevier, where it has we distribute various databases like Embase, mBiology, Reaxys, and PharmaPendium. And Embase is mostly is used for a biomedical and a pharmacological database, which covers journals, conference abstracts, drug research with a strong focus on drug safety, clinical trials, and pharmacology. And this can be used for a systematic literature reviews and regulatory research. Coming to mBiology, it's an AI-powered platform by Elsevier that helps the researchers to explore the relationship between in the drug discovery. It combines the data of scientific literature, abstracts, and the clinical studies. So it uses a knowledge-based graph to connect genes to proteins, proteins to diseases, and then the drugs. So this helps in target identification, I mean protein identification with a specific related to a specific disease, and also biomarker discovery, and also understanding the mechanism, how the drug molecule is binding and all that. We are also the distributors of SCM, which is if you wanted to do high-end quantum calculation and you want to work into the material science area, you can try using SCM. And Chemaxon, which is an alternate to ChemDraw, and it is much versatile than other tools, where you can do starting from structure sketching to Markush enumeration, library generation, and property predictions. You can create your own databases, and many more are available here. And coming to ChemPass, it's an AI-assisted lead discovery platform for reaction-based, which works on a rule-based AI, which again has 3 different modules. One is a synth space, post validator, and POVMAP. So using the synth space, it helps the scientists to quickly build the compound libraries and explore lead optimization ideas around a particular scaffold. And the scaffold hopping can be done, and side chain modifications and the retrosynthetic can also be done using this particular tool. Pose validator is again another tool which is available from ChemPass, which will help the user to identify whether the dog pose is correct or incorrect using some machine learning and deep learning other scoring functions. POVMAP, which again stands for helps in analyzing how well the library coverage is, I mean how diverse the chemical regions are being covered in that particular area. Yeah. So this is the complete landscape of MoleSoft. As we have seen, MoleSoft was founded in 1994 by Dr. Ruben Abagyan. At that time, we published 2 methodologies. One is internal coordinate mechanics and bias probability Monte Carlo. So these 2 technologies are the base or the backbone of this particular ICM. And you can see the earlier adopters, BMS and the Novartis. And the next decade, we also had very big partnerships, lots of partnerships in developing multiple tools. MoleSoft believes not only in developing tools, but we also provide solutions to our customers. Like MoleSoft collaborated with Structured Genomics Consortium for developing SCARAV and Active ICM. Again, we had a long-term partnership with Novartis for developing a tool called 3D ICM Ligand Editor, which is now extended for peptide editing as well. We also have developed a tool called atomic property fields, which is more of like pharmacophore kind of thing, this was developed by Dr. Maxim Tartov in the year 2008. And we also took part in the GPCR challenge and were ranked first commercial tool, and also took part in Capri challenge, which is more related to protein docking. So we were ranked first in this particular challenge as well. And again, in the next decade, 2011 to 12, we launched multiple other products like MoleScreen, which I discussed earlier, which has 2,500, more than 2,500 uh, modules available in that. And we also launched something called GigaScreen. And this is something, and a drug called Webomir. Rampex Pharmaceuticals launched a drug called Webomir, which is a beta lactamase covalent inhibitor, and they openly claim that the molecule, I mean, they have used MoleSoft's covalent docking tool in identifying this particular drug. And we also took part in the D3R challenges, and we were ranked first in 2 challenges, and we could again rank very better in with regards to other commercial tools as well here. So in the next decade, this being a decade of AI and GPUs, right? So we launched RIDE, which can screen up to, which is an analog based screening methodology. Even you can also consider the protein as an excluded volume here in RIDE, and we can screen up to 1.5 million conformers per second per GPU. Again, we have integrated AI based scoring function called RTCNN, and Ridge and GigaScreen, CombiRIDGE, Ginger, and various de novo tools have launched. And recently, GigaScreen version 2 has also been launched. So what are all the different modules we have in MoleSoft? Unlike other tools, MoleSoft believes in very less kind of modules. The first module what we have is ICM Pro plus homology modeling, which covers almost all kinds of structure-based uh, structure-based screening tools, starting from docking to everything. And again, we also have another module called ICM Pro homology plus VLS. And you can also independently, uh, license out ICM Chemist and ICM Chemist Pro for which will be very much useful for the medicinal chemist. So what does the ICM Pro covers? So ICM Pro covers all kinds of docking, starting from small molecule docking. And you can also do protein-protein docking and induced feed docking as well. So here an induced feed, again, there are 3 different methodologies. 4D docking, where you can, you can merge multiple conformations of the same protein. If you have a crystal structures, you can do that. Else you can generate multiple conformations by running a short MD, and then you can use this 4D docking for your further refinement of the VLS hits. So this is how you can utilize this 4D docking to enrich the data which comes out from the VLS. And we also have something called hybrid partially explicit. Again, uh, many times what happens when you have an apoprotein, let's assume a kinase. So if that being a type DFG in complex, so when you have a ligand which is a type 2 inhibitor, when we try to dock the molecule into that particular type 1 protein, which is TFG in protein, so the molecules we may not be docked correctly. So for that, either we may have to build a homology model, or we may have to do it the either other way of generating the type 2 conformation of the DFG loop. But here in MoleSoft, we have something called hybrid partial explicit, where you can select 4 to 5 residues of the loop, and you can try docking of those type 2 inhibitors, which it generates multiple conformations around that particular loop and docks your molecule into the active site. We have something called SCARE, dual scanning of alanine and refinement. So this is again when your active site is having lots of hydrophobic amino acids, where the molecules are active in real time, but using the computational tools or the docking methodology, if you are unable to understand or predict the docking conformation, so we can try using this scale, which will replace the bulkier amino acids with the alanine, and it allows your molecule to dock into that pocket. And we also integrated MD, we integrated OpenMM for the molecular dynamics. And we have also something called PROTAC modeling. And we also have something called homology modeling using ICM Pro and something called 3D ligand editor. So other than this, you can also generate 2D ligand edit, 2D ligand, 2D receptor interaction diagrams, protein modeling, electrostatic bioinformatics tools have been, a lot of bioinformatics tools have been integrated. You can also predict the effect of mutation using Mercy algorithm and direct database search. The good part of this software is direct database search. It has been, you can see here in the below, it has been already integrated with PDB search, NCBI BLAST, UniProt, PDB, sequence pattern, sequence match, sequence homologs, PubChem. So the above ones, what you are seeing are all biological databases which are integrated, and the below four, Campbell, Drug Central, crystallography, open database, and PubChem. So the user doesn't need to go out of the interface for his research. So these are all the very frequently used sites where during the research. So that's the reason these all uh, database have been already integrated into MolSoft interface. Now coming to VLS. So in the virtual screening, so the difference is you can in VL the with regards to Pro, you can dock up to maximum 100 molecules in one go. But using VLS here, there is no such restriction. You can dock millions of compounds. Using structure-based studies, you can use fragment-based studies, fragment-based VLS screening. Even you can use APF-based VLS. APF-based VLS is something where you can generate a pharmacophore of of a particular fragment and keep it in the pocket, and you can screen a database against the particular pocket where initially it screens those fragments or the molecules, and it once those compounds fits well onto this APF or the pharmacophore, then those molecules are docked. So this is a hybrid technology where other tools doesn't have this kind of technology. And this is again, if you do it independently, so we may have to generate a pharmacophore, take it out and screen it against a 3D conformation database, and then take up those molecules and then prepare those ligands and then dock them. But all these things can be done just using APF based VLS. And we also have something called ligand based VLS, which again can be done. Coming to VLS, virtual ligand screening, we have two different scoring algorithms, which is physics based and then AI based RTCNN. You can also do covalent docking, 3D pharmacophore search, and calculate the ADMET properties using AI/ML modules. What we have, hierarchical clustering can be done. 2D, 3D QSR models can be generated using VLS, and you can also. This is again a small thing. Again, there are lots of other functionalities that are available here in this ICM Pro Plus VLS. So let's quickly jump back to the ultra large library screening tools. What are all the different tools that are available with MolSoft and which are used for the the ultra large screening? Ginger, which is available both on CPU and GPU as well. Ridge is a GPU based docking engine, and Ride, which is also available on CPU and GPU as well. CombiRIDGE, there is a small mistake here. CombiRIDGE is available on GPU and GigaScreen is available on GPU. So let's quickly jump back to Ginger. What exactly is Ginger? So Ginger stands for Graph Internal Coordinate Neural Network Conformer Generator. It is a very powerful tool and GPU enabled tool which is designed for rapidly generating the 3D conformations of the molecule. And this is one of the very preliminary and very essential step in the virtual screening and the normal day-to-day activities of the drug discovery. So instead relying on the traditional slow methods, Ginger here uses neural network approach for combined with the fast energy refinement to efficiently explore the molecular conformations. So what makes Ginger stand out is its speed and scalability. It can process millions of compounds per day on a single GPU while maintaining the highest accuracy. It also generates fewer but relevant conformers, improving the downstream process for workflows like docking and virtual screening. From a practical perspective, Ginger fits seamlessly into ultra-large library scaling pipelines, working alongside with the tools like Write and Read. So again, Reach, we were discussing about Reach, which is a rapid docking GPU engine. So it is extremely fast and it can dock up to approximately 100 compounds per second on a GPU of RTX 4090, and it is 1,000 times faster than a single CPU core. And at 10 million compounds can be screened in less than 30 hours, and it is an equivalent to, this is again repeatable. So it also supports multiple GPUs. So what exactly, how this engine works? So this engine is divided into three different blocks. The first block, the CPU block, where it uses the using the Ginger, whatever the confirmations we have generated. So we consider that conformer generated database, and the first CPU blocks, it reads those conformers and it compresses the data and it assigns the charges. Means the ligand preparation is done using the CPU, and then the followed by the docking, the central part, which is again the core part, which is fully GPU driven, which does that complete docking and minimizing and hydrogen bond orientation and Cartesian relaxation. So the major task is done on the GPU. Again, in the last but not the least step, which is again nested onto a CPU, which calculates the RTCNN scoring functions, ICM VLS scoring function. This way, the bridge works. And this is something called a neural network score, which is RTCNN, radial topological convolutional network, neural network. And this was developed by considering the putative ligands that from the PDB database. And this doesn't give any kind of consider the receptor induced clashes. And you can see the predicting of the top 1, 2, and 3 uh, conformations using RTCNN. It is much better than the all other docking scoring functions. With regards to enrichment also, it outperformed a number of other docking tools. So even using Reach, what the user can do, you can do a substructure, consider a moiety of a molecule as a substructure, and you can go with the docking studies. Similarly, the one what I was saying, APF based docking. So you even using Ridge, this can be applied here. I mean, if you have a huge millions and billions of compounds, then you can try generating an APF and use Ridge, which is again very fastest way of structure based screening tool available in the market. Even this can be applied for APF based docking. And even you can also, if you have multiple receptors, which I was telling in the earlier slides, even 4D docking can also be applied to the using Ridge. And this is a recent success story with Molsoft and NIH against the the undruggable targets. So this publication explains how Ridge and Ride transform the virtual screening in the drug discovery. So Ridge is again fast, accurate, structure-based method which I've been telling about. The truly exciting part about this research paper is the application of Write and Reach against very challenging targets like PD-L1 and KRAS G12D. So despite being difficult to drug, this study we could successfully identify novel inhibitors with a stronger activity. In a very simple terms, this work shows how GPU acceleration can now explore previously unreachable chemical space and discover a meaningful candidates faster than ever before. So coming to RIDE. So RIDE again similar to RIDGE. RIDE also uses pre-generated conformers, which is a rigid body APF superposition using optimizes using translations and rotations. So this is much faster on C, not much faster, even good speed. It has a good speed on CPU and especially on a multi-core, and it is very faster on the GPU. So what are all the applications? It can be used for virtual screening, where you can efficiently screen giga size library. You can also use this for scaffold hopping studies and hit follow-up, and you can also give certain weights with regards to the specification of the pharmacophoric features which you wanted to extract the output. You can also add protein as an excluded volume using this in this particular reach. And how this right, what in the back end it considers, APF. So what are all the different features this APF does it consider? It's a 3D pharmacophore model potentially that can be used for chemical superimposition, multi-compound alignment, virtual screening, 3D QSAR, and it has various features like hydrogen bond donors, hydrogen bond acceptors, sp2 hybridized, lipophilic, and the size small lipophilic areas and positive and negative charges. And what are all these are the benchmarking studies as well as the different libraries what you get once you have a GPU based RIDE license. So you can get the libraries of from MolSoft Molcart with around 10 million compounds, and eMolecules 27 mid-sized libraries like MQL, Enamine, and giga-sized libraries Wuxi, Savvy, synthesizable, and Enamine Real, and these are all can be provided once if you have this RIDE GPU license. And with regards to the speed benchmarking, so these are all the some of those things. A flexible APF takes around 44 days to screen a database of 1.5 million compounds with 30 confirmations average, and the rigid APF would take around 22 days to screen this database, whereas the same database was screened using RIDE CPU. It took 1.7 hours, around 229 compounds per molecules per second, and RIDE on GPU is it can screen up to 38,729. So this is how the RIDE GPU and CPU are much faster than the traditional APF, flexible and the rigid APF methodologies. Yeah, let's coming back to the CombiRIDGE. So CombiRIDGE is again a GPU accelerated platform for high throughput docking. But and again, CombiRIDGE is an integration of three technologies: Ginger, Ridge, and the fragments which are or the compounds which are identified are scored using RTCNN, which is a graph neural network, which is an AI-based scoring function again here. So here in this methodology, it's an anchor-driven approach where it starts with it docks the fragments into a particular pocket, and once it identifies the crucial interactions, that fragment is extended with multiple R groups on that particular fragment. So followed by the efficient optimization of various R groups screening. So this is one of and the database that is available for this particular CombiRIDGE is from Enamine, which consists of a 45 million anchor library size, and Wuxi Galaxy, which has a 35 million anchor library size, and eMolecules, which has around 60 million. So most of these are like small molecules, I mean less than 300 molecular weight fragments kind of things. And this is again the same methodology which was the benchmarking was done with regards to speed. So this was done on a RTX 4090, where 28 billion chemicals from Enamine real database were screened. In the very first step, it is the anchor processing, and then followed by the second step is the 3D conformation generation, where approximately 18 million anchors molecules at a speed of 7,500 fragments per minute, which took around 40 hours to complete this computation. And the third step is anchor docking using RIDGE, which is again very ultra fast docking, which took around at a speed of 3,500 fragments per or the anchors per minute, which took around 90 hours to complete the computation. So again, the in situ optimization using Ginger and GPU accelerated MMFF minimization, which took around. So this way, it took around 106, 136 or close to 140 hours to complete this computation. And CombiRIDGE, and this is a test case where what you are seeing. Let's take an example of the Veneto class. So in the it is a first class drug that targets BCL2 protein. So that BCL2 is a protein that the cancer cells use to avoid dying. So in many cancers, due to the overexpression of BCL2, it helps the tumor cells to survive longer than they should be. So venetoclax works by blocking this particular protein BCL2, and then you can see the molecular weight. It has like around 868 kilodaltons, which is much bigger than a small molecule. So in this study was started by docking of 1 million small fragments and a hypothetical building block of venetoclax. So through docking and the smart selection and iterative design using CombiRIDGE and Ginger, we were able to construct this particular molecule. So in simple terms, complex molecules like venetoclax can be rediscovered using this approach. Yes. Coming back to GigaScreen, again, instead, this is an AI assisted, this is a deep learning approach which Molsoft has. So usually when you have very huge libraries, instead trying to dock the complete database, so it is we here GigaScreen tries to model and instead docking the entire massive database at once. GigaScreen takes a smarter and iterative approach. What it does, the process starts by selecting a very small subset of compounds, about 0.1% of the database, which typically comes around 1 to 2 million compounds. From the beginning, the data is again split into two. You can see data is split into two: training and the test set. And the GigaScreen uses the techniques like the fingerprint techniques like ECFP, extended connectivity fingerprints, and also GCNN models. So the predictive model is again trained on this particular substrate. Once trained, the model is applied to the entire database to identify the most promising candidates. So a specific cutoff of again, it is user defined. A cutoff of 80% would be better. So ideally capturing 80% best hits. If the data set is too large, so we can keep repeating the process with a new subsets gradually, which refining the model. So over a time, the model improves and the scores get better and the number of compounds narrows down, and to get a very focused high quality data set. So for a 2 billion molecules database with approximately 5 iterations for the training and test set and everything backend processing, it takes around approximately 72 hours. For each iteration, it takes around 15 hours, and the complete computation, like it takes around 72 to 96 hours to complete the docking of these 2 billion compounds using deep learning models. So this is what we have for today's webinar session. So with this, I would like to conclude the session and I'm open to questions.
Thank you so much, Dr. Sardar, for that insightful and highly informative session. Now we'll move to the Q&A session. So the first question we received is, will we get ultra large screening tools with ICM Plus Pro VLS license?
Sorry, can you repeat?
Yes. Will we get ultra large screening tools with ICM Plus Pro VLS license?
No, No, these are the GPU and the modules what we have seen are not the part of your either ICM Pro homology or ICM Pro plus VLS. They need to be licensed separately.
Okay, sir, got it. And sir, second question we received is, can we use multiple GPU for these tools?
Yes, definitely. Multiple GPUs can be used.
Okay, sir, got it. And sir, thank you all audience for their excellent questions. And your active participation truly makes this session more engaging and valuable. While we try to address as many queries as possible, a few may remain unanswered due to time constraint. So our team will get back to you via email with detailed response. With that, we came to the end of today's webinar. On behalf of Advent Informatics, I extend a sincere thank you to Dr. Samsher Singh Sardar for sharing his expertise and valuable insight on this advanced topic. And big thank you to all our participants for your time, for your engagement and support. So we look forward to welcoming you again in our upcoming webinar with that more insightful sessions. Till that, have a wonderful day ahead. Thank you so much.
Thank you, Sailesh. Thank you, everyone.
Thank you, sir.